So a lot of research being done on board the International Space Station, actually taking a look at crew health and how the human body is able to adapt to the rigors of spaceflight. The SpaceX Dragon vehicle, the sixth resupply mission for SpaceX, is bringing up about 2 tons of new research. And inside is a pretty exciting new study called Osteo4. And today I'm joined by the experiment's principal investigator, Dr. Paola Divieti Pajevic. Coming to us. She's an associate professor for molecular and cell biology from Boston University. First off, doctor, thank you so much for joining me today. And if you could start off, so what is it your experiment's looking at? I hear it's something called osteocytes. Can you walk us a little bit through what that is and just a general overview? What are you guys going to be looking at?
Sure. Well, first of all, thanks, Dan, for having me here. So the osteophore is osteocytes and mechanotransduction. And osteocytes, for whoever is not really familiar with bone biology, are the most abundant cells in the bone. And these are the cells, they are completely embedded within the mineralized matrix of the bone. And these are the cells that you thought that they can sense the mechanical forces or the load applied to the skeleton and then transform this mechanical load into biological signal. So it's known that during reduced load, like during, for example, prolonged bed rest or during a space flight, there is a bone loss. And the bone loss is mostly driven by signals sent to bone cells capable of making bone or destroying bone by osteocytes. So the goal of the osteophore mission is really to identify genes and proteins or factors that change in osteocytes by microgravity. And this will be the first time that osteocytes will be flown in space. And this project is funded both by NIAMS and NIH, and also funded by CASIS and by the NASA Kennedy Space Center.
Okay, so we're looking at osteocytes. You said they're the most abundant cell, and they sense things like mechanical forces. So, things like weightlifting, I assume, are something they set off. What are the actual operations that you guys are going to be doing to get this data? What's physically going to be happening in this experiment on board the station?
Right. So, so we will use an osteocytic cell line. We will use a cell line that we isolated, we developed in my laboratory. And these cell lines are developed, were isolated from long bone of mice. These cells will be grown in a three-dimensional structure. It's a 200 micron thick structure that mimic a little bit more the three-dimensional structure of bone. And we will house these cells in scaffold, and the scaffold will be integrated in bioreactor. Bioreactor are fully enclosed system in which we can grow the cells because there will be nutrients provided to the cells during the mission. And then this bioreactor will be integrated into a fully automated fluid pathway that will provide not only the nutrients to the cells, but also later time will provide fixative, and then we can preserve the cells for a later study. So during the mission, we have 9 bioreactors in this mission, and at different time along the experiment, astronaut will retrieve this payload in which there will be 3 bioreactors, and we will store them at cold temperature. And then once the experiment will be done, and we have operation happening at right almost as soon as the Dragon reach the ISS. So we will have operation happening after 3 days or 5 days or 7 days that these cells have been exposed to microgravity. They will be retrieved, stored at cold temperature, and then the samples will be brought back by the same vessel, by the Dragon, and then they will be brought back to my laboratory, and then we will study them once they are back in the lab. And this actually, the system was developed by CAM Technology in collaboration with the Canadian Space Agency, and this actually is the 4th time that this payload is flying. And also actually is an acronym for osteoporosis experiment in orbit. But this is the first time that the payload will be actually physically brought into the ISS.
Okay, very, very cool. So what are some of the next steps after you get these samples back, from orbit? Dragon's going to bring them home, and you said they're going to come back to you. What are some of the next steps for the experiment?
Well, definitely we'll be very careful when we use or when we analyze the samples. Mm-hmm. Well, what we will do, we will both isolate RNA to look for gene expression. We will try to do gene profiling to try to identify either genes that we know are changed during disuse or microgravity, but also try to see if we can identify novel targets. And then we will also look at the cell structure, how the cells are changing, and we also look at some protein expression. We might do proteomic, that is like a just screen for all possible protein that are changed in these cells, and then try to identify possibly potential novel target or novel genes that we can target for down the line, maybe develop novel therapeutic for osteopenia or osteoporosis or bone disease.
Okay, and just to, bring it all home for us, what are some of the benefits that we could reap from a study like this? What could this improve for not only, people in space, but people down here on the Earth?
Correct. So the mechanism of bone loss that happen when you are in space are the same mechanisms that happen when you are, for example, a prolonged bed rest, or if you are during paralysis, or just during old age, in which there is a bone loss. So try to study a system, like in our case, osteocytes, in an extreme condition like in space. We really give us a pretty good clue of what's happened to the bone for when here on Earth. So will not only give us some novel pathway or potential therapeutic for treating bone loss due to disuse, like in space flight or bed rest, but also potentially help us to treat any kind of bone disease that will lead to like reduced bone mass, like just aging or osteoporosis, that affect like 5 million or even more people on Earth. So I think that we will use the ISS as our national laboratory in space to really try to benefit the people on Earth.
All right. Well, a very, very interesting study, some really interesting stuff that could come out of it. And, we're so glad to hear about it. So again, Dr. Paola Divieti from the Boston University, the principal investigator of Osteo4, just a couple hours away now from the International Space Station. Thank you so much for joining me today, giving us an inside look at your experiment. We really appreciate it.
Yeah, thanks a lot for having me.